Okay, this is our second last topic to the end of biology. And this is homeostasis that I'm going to cover in 2 videos. We're going to start with a PowerPoint that I have sent to you. You'll need your student notes and your question pack to work alongside this, and some paper as well. First task is a match up. You'll need your keynotes to complete this. So there are 4 key terms, 2 which you're already familiar with from the nervous system, receptor and effector, but 2 brand new key terms, positive feedback and negative feedback. In the past we have had feedback and its importance in biology as an essay title. The only place we've met positive feedback so far has been in the nervous system, when the voltage gated sodium ion channels opened and caused more sodium gated ion channels to open. So that might give you a bit of a clue. Okay, have I got matching up? Pause here and then catch me up. Okay, so you should have completed this now. So positive feedback then, in terms of a definition, we're looking for a movement away from the norm produces a response that takes the value even further away. So C. So this idea that in the sodium channels, when they opened, the sodium moved in, it caused more sodium channels to open, so more sodium moved in, so the depolarization got bigger and bigger and bigger. A receptor, well, this is, whoops, that should be up here, there should be the thing that detects any deviation from the set point. So we'll ignore that and we'll just put D there, please. Okay, so negative feedback then. So this is a movement away from the norm produces a response that returns the value to norm, so that should be an A there. And finally, effector is B, what is left, brings about the response which returns the conditions back to norm. So in the homeostasis topic, we'll be talking really about negative feedback and how receptors detect changes away from the norm, and effectors then return the conditions back to the norm. Positive feedback, really the only example that you need to know about is within temperature feedback, because it's a nice synoptic one. So if you think about when somebody is really super cold, one of the things that you do to heat yourself up when you're really cold is shiver. But when the temperature drops so far, you enter a positive feedback loop with hypothermia, because you are so cold you no longer can shiver very well, and because you can't shiver very well, you get colder, and then you can't shiver even more, less well, and so on and so on, until you can't return your body to normal temperature. Okay, so temperature control is probably the one that you have met for homeostasis. So into your keynotes for a quick review of those key terms. Okay, so in your keynotes, you've got your negative feedback here and your positive feedback here. And the example that I've put in your keynotes for your positive feedback is the one I was talking about with the temperature drop in hypothermia, but you might want to add in the sodium channel example I said in the starter. Everything we're going to look at in this topic is a negative feedback mechanism, so we're looking at ways in which the body restores systems back to their original level. So that means when there is a change away from the set point, the set point being the normal temperature, the normal glucose concentration, the normal pH of the body tissues, then things happen, effectors bring about changes that return to that set point. And of course, receptors are what detect the change in the first place. So we're going to look at some examples of that. Okay, so that is basically our homeostasis topic. So in this bottom bit over here, you can see that I've also defined receptors as things detecting stimuli. So that's the same as we've met previously. Effectors in this case bring about responses, but what's slightly different is that the response is like a corrective measure. So in other words, it returns to the original condition. The coordinator is just the brain. We don't need to know any particular regions yet. We will for specific examples. Okay. And the set point is what is also called the norm or the desired level. So that could be a particular body temperature, could be a glucose concentration, it could be a pH. Okay, so those are kind of generic bits in your keynotes, which sometimes the examiner sets exam questions on mechanisms you haven't met before. But because you know these keywords and what they mean, you can then apply your knowledge. And we'll see a couple of examples of that in the question pack. So back into the PowerPoint, and you're just working on this first section here in keynotes. So we're thinking now what homeostasis is, what this topic is about. Okay, so it has definition. You'll remember it from GCSE. Homeostasis is the maintenance of a constant internal environment. So think of the things that you learned when you were doing your GCSE homeostasis. Cells work best if they have the correct what? What kind of factors did you talk about controlling in homeostasis at GCSE? I'm hoping they're the ones you've come up with. So temperature, water levels, and glucose concentration. You might have also put in the pH, which is a way of controlling solute concentration. You might think as well water levels. And that would be it. Okay, so there's a synoptic link here. I'd like you to have a little think about your knowledge of enzymes and their sensitivities when it comes to pH and temperature changes, because sometimes the examiner will say, why is it important that homeostasis maintains the set point temperature or pH for a particular organism? And the answer would always be about enzymes. So explain these the effect of these factors. I'll give you 4 marks, just 4 minutes to do. If you pause there and give it a go. Okay, so thinking about pH first of all. So a change away from the optimum, that's in either direction, acidic or alkali, results in the enzyme losing its tertiary structure. So that means hydrogen and ionic bonds are broken between the R groups, and its active site is no longer complementary to the substrate, and the rate of reaction, or whatever reaction it catalyzes, slows and stops. Temperature is just slightly different. For temperature, it's just above optimum temperature that this effect is felt. High kinetic energy breaks the hydrogen and ionic bonds, again between the R groups, resulting in the loss of tertiary structure. So its active site is no longer complementary to the substrate, and the rate of reaction it catalyzes slows or stops. So that's just applying your knowledge. So in this topic, 1, you might need to define what a homeostasis is. So you want that simple definition. 2, you might have to apply some of the key language like receptors, effectors, and negative feedback to unknown homeostasis mechanisms. Temperature is one you've done at GCSE, so we will recap it, but it isn't on the specification in any detail. And then we're going to introduce your AO1 one, which is blood glucose, and that one you do need to know in an awful lot of detail. Okay, so if we think about temperature, this is the one you know from GCSE. Just stop here and write down everything you can remember about temperature control and homeostasis from your GCSE knowledge. Okay, right, should be back with me. Let's see what you could remember. So most people know that normal body temperature or set point is 37 degrees C. Okay, so what happens if your body temperature goes above that? Well, if your body temperature rises, then the receptors in your body for temperature will be able to bring about some changes in your body. So there will be receptors that detect the change, a coordinator that brings about the change, and then effectors. And the effectors include things like causing your blood vessels to dilate, causing your sweat glands to secrete sweat. All of those effects will cause an increase in the increase in heat loss to the surroundings by your body, and so lower your body temperature. If your body temperature were to drop below 37 degrees, again, receptors detect that change. The coordinator, the brain, brings about a response via effectors, and these are the responses you remember from GCSE. They cause constriction of blood vessels, so that blood flows under the fat, and it's insulated, and therefore you lose less heat across your body. The sweat glands remain inactive, and we can also cause involuntary muscle contraction, so in other words shivering. These two effects, reducing heat loss from the body and increasing production of heat as a waste product during respiration from extra muscle activity, cause an increase in temperature. So that's the one that the examiner expects you to be familiar with. The control point for temperature is found in the hypothalamus. Okay, so that's where we'll monitor the receptors are also found in that region of the brain. Okay, so there may be, if they did ask you about temperature, it will be more of a synoptic question. So why be warm-blooded? Why to go to all this trouble to maintain a core body temperature? Because it's only organisms like ourselves that are warm-blooded that do this. And if you think about it, what animal B, with its homeostatic control of its temperature, is able to keep its temperature constant, whereas animal A, you can see it's cold-blooded and its temperature fluctuates in line with the environment. If you think about catalysing reactions, rates of reactions, keeping the body temperature up near optimum temperature for our enzymes means that we are catalysing reactions at the fastest rate, the most optimum rate. We don't drop lower than that or go above it. Whereas organisms that are cold-blooded will be very temperature dependent on the environment. So that when the body temperature, when the environmental temperature drops, the body temperature drops, and so do their rates of reaction. Okay, so we are going to have a look at the homeostatic control, which is our focus point for the A-level course, which is control of blood glucose. Now, to do this, I would like you to listen to the clip that I have sent along with this link to this video in the email that I've sent you. Okay, it's a really strange song. It's a bit of a laugh. It's control of blood glucose. It talks about the organ which controls blood glucose. So these are the questions. What organ controls blood glucose? Does this organ do anything else? And which hormones control blood glucose? So if you listen to that song, okay, if you listen to it, it is a bizarre song, I know, but you should have picked up that it's the pancreas that controls blood glucose. Okay, it also secretes enzymes and hormones too. So there was a couple of other things mentioned in the song, and the hormones that control blood glucose were insulin and glucagon, so you would have heard that in the video. Okay, so if we have a look at the pancreas, our super pancreas that we love, you can see that pancreas connects up to your small intestine via the pancreatic duct. This tiny duct allows the secretions from the pancreas, including these hormones, to make their way, sorry, not including these hormones, but the enzymes that we talked about in year one that are important in digestion to leave the pancreas and move into the small intestine. The hormones that we're talking about in this topic leave the pancreas in the bloodstream. So you'll see that if we do a cross section of one part of the pancreas, there's many capillaries for these hormones to move into. And that's the thing about hormones and the endocrine system, as you can see in the keynotes. Blood glucose is a negative feedback mechanism controlled by the endocrine system. So this is where hormones are released. Okay, they're produced in glands and secreted into the blood. You'll remember that from GCSE. They're then carried in the blood to the target cells, which have receptors on the cell surface membrane. They're complementary to the hormone, and the hormone binds to them and affects that target cell. And they have, long-lasting effects, and are often widespread in their effect. As long as the target cell has got the receptor, it will respond to the hormone. Okay, so back to our pancreas. So if we were to look at the pancreatic tissue, it's built up of many specialised pockets called islets of Langerhans. Okay, in the islets of Langerhans, you can see we've got lots of capillaries, so that's when these cells produce the hormones that control blood glucose. They are secreted into the blood by those capillary systems. Then we've got the alpha cells around the outside of the island and the beta cells making up the middle of the island. The alpha cells, they secrete glucagon and the beta cells insulin. Can you remember when these cells secrete these hormones from your GCSE knowledge? Okay, so I'm hoping you've remembered that glucagon is secreted when your blood sugar drops. Okay, so say you've not eaten for a while and your blood sugar starts to drop. It's the alpha cells, which were around the outside of the islets of Langerhans, that secrete glucagon into your blood. That glucagon then travels in your blood, binds to receptors on target cells, and brings about an increase in blood glucose. So that is a negative feedback loop. We've detected a drop in blood sugar. The receptors have caused the pancreas to secrete glucagon, and the glucagon has then bound to target cells, causing glucose to be released into the blood. In contrast then, I'm hoping you remembered that beta cells, they secrete insulin when your blood glucose goes up. So just after eating, you'll secrete insulin into your blood. That insulin will travel in your blood and it will bind to receptors which cause the glucose to be uptaken from the blood by those target cells. So these cell types, we can say, have an antagonistic action. So we've met this word now in 3 topics. Okay, so we met it with the parasympathetic and the sympathetic arms of the autonomic nervous system. We met it with the bicep and tricep action in the arm in antagonistic muscles. So what did it mean? Okay, so it's that idea that they have opposite actions. So glucagon is released in response to a drop in blood sugar and brings about an increase in blood sugar, whereas insulin is released in response to an increase in blood sugar and it brings about a decrease in blood glucose. Okay, quick review. Name the site of glucagon synthesis. So where exactly, precisely, was glucagon produced? So saying the pancreas wouldn't be good enough, saying the islets of Langerhans wouldn't be good enough, we'd have to say the alpha cells of the islets of Langerhans of the pancreas. Whereas for the site of insulin production, B, we would be saying the beta cells of the islets of Langerhans of the pancreas. So what prompts the release of insulin from the pancreas? We said that would be the detection by receptors of an increase in blood glucose as its response that it coordinates would be to return that blood glucose back down. In contrast, how does insulin cause the blood glucose level to decrease? Sorry, not in contrast. How does it do that? We said it brings about responses where glucose is removed from the blood, but we'll have to see what they are next. Okay, so you've got quite a bit of work to do now. I'd like you to use the whole of page 5 and 6 and 7 to complete this task. That's 5, 6 and 7 in your keynotes. Okay, I want you to get some A4 paper and I want you to focus on those keywords and I want you to draw a diagram that illustrates the control of blood glucose. Okay, if you're struggling a little bit, then you can play and I will do it alongside you, but if you can give it a go yourself first, that would be brilliant. Okay, so hopefully by now you'll have your paper. Alright, so what we're going to do is we're going to separate the paper in the middle and call that our set point. Okay, so we're talking about between 80 and 100 micrograms per 100 centimetres cubed of blood. So that's the normal glucose level of a healthy person. So that would be our set points. Our homeostatic control mechanisms will try to return our blood sugar to this level following a meal or following fasting. Okay, so we want to see how that's brought about. So if we think about an increase in blood sugar and a decrease in blood sugar. So we do increase, I keep saying blood sugar when I should say blood glucose, increase in blood glucose. It's just when people talk about blood sugar, say diabetics, etc., they are talking about blood glucose. Oh no, I've written increase again instead of decrease. So I should say decrease at the bottom in blood glucose. Okay, so we've got both things that can change. So an increase might be, i.e. a meal, so you've just eaten and digested your food. That might be an example of a decrease in blood sugar would be following a period of fasting. Okay, so the first thing, both would be detected by receptors in the body. Okay, and that would then cause a coordination of a response, and it's our pancreas that is the response, or controlling the response. So if we've got an increase in blood sugar, that would mean that our beta cells in the pancreas would be bringing about the response. So they would be the effector cells, if you like. And they would be doing that by secreting insulin. Okay, so they will secrete insulin. And the insulin will travel in the blood from the pancreas to the target cells in the body. When it gets the target cells, it will bind to receptors on the surface of the target cells. And it brings about a number of responses inside those cells. So one response is that it causes the cell to introduce more proteins for the uptake of glucose in the cell membrane. So that means that there are more transport proteins for glucose in the cell membrane, and that will cause an increase in glucose uptake. Okay. So insulin will bind to receptors on target cells, and inside those cells it will cause them to produce more of these transport proteins and put more of these transport proteins in the cell membrane, so that that cell will now take up more glucose. That will start to lower the glucose in the blood. At the same time, inside the cells, it activates enzymes, and it activates enzymes which convert glucose, the monomer, to its polymer storage molecule, glycogen. Now we have to be careful with this now when we write glycogen, because it's got to have an E there, it's got to look like an E, because we've now got glucagon as well, and the examiner gets really annoyed with handwriting like mine. Okay, so it activates these enzymes, that catalyse that reaction. So you get more storage of glucose. So there's going to be more glucose taken up out of the blood and more glucose converted to glycogen inside those cells. So overall, this is going to cause the blood glucose level to drop again. And return us to our set point value. So this is one negative feedback loop. Yeah, we've had one increase in blood glucose followed by a response that has returned the blood glucose concentration to normal again. So it's a negative feedback loop. Okay, we have the same thing for a decrease in blood glucose. Okay, detected by receptors in the body, brings about a response by our alpha cells this time in the pancreas, and it causes them to secrete. Now we have to be careful and make sure you spell it correctly. Okay, glucagon. So we've got glycogen and glucagon and lots of other glies and stuff that's going to come soon, so just be really cautious. Okay, so glucagon being our hormone, it binds to receptors on target cells, but brings about a number of responses. It causes the breakdown of glycogen back into glucose by those cells, and that glucose then moves into the blood. Under some circumstances, for example, if you've not eaten for a long time, you might start to run out of glycogen stores. So it also activates enzymes that do some really special stuff, converting amino acids and also some glycerol into glucose, and that glucose can end up in the blood then. So both of those actions mean that the blood glucose level would increase. Okay, so that's another negative feedback loop. Okay, so in the keywords that I gave you, there were some keywords that we haven't used yet. I'm just going to take a different pen colour. Okay, so, and it's the names of these that are so important to the examiner, these responses. They all have special names. Okay, and I've also given you the name of the disease states if the blood glucose dropped too low or went even too high, so that this mechanism wasn't working. So the consequence of those in both cases is death. So this is here and here. Okay, so we'll start with putting them on. Okay, so if this were not to happen, if you didn't detect that glucose increase and the beta cells didn't secrete insulin, so I think you'll think of a particular disease that might happen in, when people might not be able to secrete or make insulin, so this response wouldn't happen, then one of the things that might happen is this process. The blood level of glucose may become too high, and there will be in a disease state called hyperglycemia. So the hyperglycemic coma, for example, you might have heard of. So they've gone hyper. So that's following a meal. Okay, not being able to secrete insulin. The other extreme is a hypo, when your blood glucose goes too low, and that still does happen to people with diabetes. So hypoglycemia is the disease state where your blood glucose drops really low, below normal. Okay, so the other things that we should have on, which are these three really important reactions, if you like, names for. So being able to make make glycogen from glucose, so making glycogen, is called glycogenesis. Okay, so we're making glycogen, glycogenesis. So that's what happens with insulin when glucose is converted, joined by condensation reaction, many of them, to make glycogen. Okay, glycogenesis. Whereas we've got the opposite reaction here, which is called glyco again. This time glycogen, so you've still got glycogen. This time we're breaking it down, so it's oolysis. Glycogenolysis. So both of them are talking about glycogen, so you can see glycogen in their name. So it's glycogena. So I always got glycogenesis. So glycogen is hidden in the name, and glycogenolysis. So again, glycogen is hidden in the name. The one that stands out as being a bit different is this process of converting amino acids and glycerol into glucose. This time we're not making glycogen, we're making glucose. And this is called gluconeogenesis. So for me, we're making glucose now. It's brand new from scratch because we're not starting with anything that's like a carbohydrate, and we're making it genesis. Okay, right, so they're the three key terms that are new and very A level, and they're very much like those three. Okay, so back to the PowerPoint. Okay, so in the PowerPoint you should be able to match up now these reactions. Now the target cells that I've been referring to, a lot of them are liver cells. So the liver and muscle are really the target cells for insulin and glucagon. So can you match up these three key terms with the reactions? I've also put in red the name of the enzymes, which is off spec, just for essays really and research. Okay, so have you started to match them up? So the first one is glycogen, you can see in there, so glycogenosis. So that was making glycogen. So you should have matched that up with C. Okay, so glycogen synthase is the enzyme that catalyzes that reaction. That's not too hard to remember. For this one, remember now it's gluconeogenesis. So glucose, new, and generating it. So that should tell you then that this is the conversion of glycerol and amino acids into glucose, leaving us only glycogenolysis as A, the conversion, no, not A, sorry, B, the hydrolysis of glycogen to glucose. So glycogenolysis, hydrolyzing glycogen to glucose. And the name of this enzyme is glycogen phosphorylase. We might meet that again. Okay, so that's where we're going to get to for now. We're going to do a quick whiteboard quiz now. So you won't be able to necessarily do the whiteboard because you might not have one at home, but you can write down the answers you think on the paper in front of you. So, first one, define negative feedback and give an example. You can pause at these in between so I don't need to stop and wait. Okay. So, negative feedback. This is where a change brings about responses which return the conditions back to the optimum or the normal set point. And insulin release after food is a good example. What about positive feedback? This is where a response brings about responses which take the conditions further away from optimum. So I gave you two examples. Sodium ion channels was the one I've just given you. So them opening, causing more sodium ion channels to open, and so depolarization. I explain why blood glucose must be controlled. To ensure glucose is available for all cells for respiration, and to maintain the water potential of a tissue fluid, so excess water is not lost or gained by cells, which could damage them. Both synoptic ideas, both things they could ask you in the exam. How does the body return blood glucose to set point following a fasting period? So this is the kind of clue you'd look out for in the exam questions. Fasting, straight away you'd be thinking, well, blood sugar would be decreasing. That would be a glucagon response. So have a go at writing the response down in bullet points now. So you should have a low blood glucose is detected by receptors. They're found in the pancreas. Glucagon is released from the alpha cells in the islets of Langerhans. Glucagon binds to receptor proteins on the surface of target cells, particularly the liver, and cause glycogenolysis, so that activates glycogen phosphorylase, so it's breaking, hydrolyzing glycogen down into glucose, and the glucose goes back into the blood to raise the blood glucose level. And at the same time, it causes activation of the enzymes which convert amino acids and glycerol into glucose, producing new glucose, so gluconeogenesis. Next one, how does the body return the blood glucose to set point following digestion and absorption of a meal? This is usually the one that comes up on the exam the most. So high blood glucose, again, detected by receptors in the pancreas. Insulin is then released from the beta cells in the islets of Langerhans in the pancreas. Binds to receptor proteins on target cells, liver particularly. Now, more glucose carrier proteins are placed into the cell membrane, so the cells become more permeable to glucose and take more up. And at the same time, it causes glyconeogenesis, activating glycogen synthase, which means the glucose is joined by condensation reaction to the glucoses to make glycogen and to store it in the cell, taking more glucose out of the blood. Okay, so we're going to stop there and let you digest this a little bit. And then we're going to move on to the more difficult aspects of this one, which is the secondary messenger model. So it's how does the glucagon bring about those effects inside the cell, and that's the same effect as adrenaline. So we're going to have a look at that in our next video. Thank you.